Hello, everyone. It's my honor to introduce a paper our team recently published in the journal iMeta, titled Metagenomic Analysis Sheds Light on the Mixotrophic Lifestyle of Bacterial Phylum Zhuqueibacterota. Lian Zheng-Han from Sun Yat-sen University is the first author, and Professor Wen-Jun, Dr. Lian, and Dr. Jiao are the corresponding authors. As we all know, the Earth is home to a vast biodiversity of prokaryotic microorganisms. However, only a small fraction of bacteria can be cultured under laboratory conditions. KSB1, an uncultured group, was first identified in sulfur-rich marine sediments through 16S rRNA gene clone sequencing. Since then, KSB1 has been found to be widely distributed across various habitats, including marine coastal areas, microbial mats, cave sediments, aquifers, and pig manure sludge, and it may be particularly enriched in anoxic environments. Phylogenetic analysis reveals that KSB1 is divided into 4 distinct clades, each characterized by unique gene profiles and niche adaptations. Metabolic reconstruction of these K1 clades indicates an anaerobic heterotrophic lifestyle, which highlights the differences in their potential ecological roles. However, the limited number of high-quality MAGs has constrained our ability to explore their ecological functions and evolutionary history more deeply. We have conducted a long-term sample collection of hot spring sediments from Tingchong since 2016, and a total of 17 sediment samples were used in this study. We performed ultra-deep metagenomic sequencing on each sample, with a minimum of 30 gigabase pairs per sample. This process yielded a total of 650 gigabase pairs paired-end reads. Through metagenome assembly and genome binning, with 30 high-quality KSB1 MAGs. Subsequently, we downloaded 45 high-quality MAGs from public databases for downstream analysis. Based on phylogenetic analysis, KSB1 forms a monophyletic clade within the FCB superphylum. This clade can be further divided into 5 distinct groups, which correspond to 5 orders of the undescribed phylum KSB1, as recognized in the GTDB classification system. From our analysis of 75 MAGs, we have identified 1 class, 5 orders, 14 fam, 27 genera, and 40 species. Consequently, we propose the name Zhuqueibacterota to encompass this newly characterized group. Through the IMNGS web server, we aligned the 16S rRNA gene to the amplicon dataset in the SRA database. We discovered that Zhuqueibacterota exhibits a global distribution, with a strong presence in soil samples, and it thrives in biofilm and hydrothermal environments. Moreover, distinct lineages exhibit distinct environmental preferences. We reconstructed the metabolic models for each order with functional annotations. Zhuqueibacterota possesses diverse metabolic potential, including near-complete central carbon metabolism and the capacity for anaerobic fermentation. Moreover, there are significant functional differences between different lineages. Order 1 and Order 5 encode multiple cytochrome C oxidases, suggesting that they are more to be facultative anaerobes rather than obligate anaerobes. Surprisingly, we found that the RBCL gene is ubiquitously encoded in Order 1, and phylogenetic analysis has identified it as type I, which is responsible for carbon fixation via the CBB cycle. Concurrently, we detected a gene cluster related to the CBB cycle upstream and downstream of RBCL, further supporting the idea that it possesses carbon fixation ability. Among the potential fixation species, we discovered the coexistence of membrane-bound hydrogenase, soluble hydrogenase, and hydrogenase maturation proteins. Zhuqueibacterials may utilize hydrogen oxidation to supply energy and reducing equivalents necessary for carbon fixation. Based on this, Zhuqueibacterials could potentially be a chemoautotrophic hydrogen oxidizing bacterium. Zhuqueibacterota exhibits a mixotrophic lifestyle. We predicted gene gain and loss events and reconstructed the ancestral genome content. Over time, contemporary Zhuqueibacterota have acquired more gene families. Frequent horizontal gene transfer events occurred in Order 1, particularly in MAGs associated with hot springs. The SID-AB gene was acquired early on, while the COX-ABC gene was independently acquired by different lineages. Genes related to anaerobic fermentation were lost at multiple evolutionary nodes. Zhuqueibacterota inhabiting thermal environments have gained the ability for carbon fixation, which endows them with a mixotrophic lifestyle. In summary, in this study, we proposed the classification of Zhuqueibacterota, based on hot spring metagenomes and public MAGs, within the FCB superphylum. This new group comprises 1 class and 5 orders. Zhuqueibacterota is globally distributed, with the majority of its members being facultative anaerobes. Notably, Order 1 utilizes hydrogen as an electron donor carbon fixation via the CBB cycle. Phylogenetic and metabolic analyses underscore Zhuqueibacterota's critical role in the carbon cycle and the prevalence of frequent horizontal gene transfer events throughout its evolutionary history.